Hello everyone, and welcome to Lab Manager's Tech Trends webinar series. My name is Trevor Henderson, Technology Editor for Lab Manager, and I'll be moderating today's discussion, which will focus on trends in drug discovery technology. Today we have a panel consisting of two leading experts in the field who have joined us to discuss some of the latest technology trends in the exciting and rapidly changing drug discovery field. We like our webinars to be very interactive, so we encourage you to send us your questions at any point during the presentation, and the panelists will address these during the question and answer session following the presentation. To ask a question, simply type your query into the Q&A box located at the bottom left-hand side of your screen. We'll try to address as many questions as possible during the question and answer session, and if we happen to run out of time, I will forward any unanswered questions to the panelists, and they can respond to you via email. You may also move or resize any of the windows simply by grabbing them at the top or stretching them at the bottom right corner. At this time, you may need to move or minimize some of the open windows to see the live view. This webinar recording will be available early next week on Lab Manager's website. At the end of the webinar, we will share that link with you, as well as the contact information for all of our panelists. With that, let me introduce our first speaker who joins us today from Thermo Fisher Scientific. Keely Murphy is currently a Senior Marketing Specialist at Thermo Fisher Scientific in support of pharma drug discovery workflow and application development, and focusing on triple quadrupole and high-resolution mass spectrometry for the quantitative analysis of bioanalytical assays. Prior to joining Thermo Scientific, Keely was responsible for workflow development of LC-MS analysis quantitation for the early ADME assays at the Novartis Institute for Biomedical Research, and group leader of the LC-MS Quality Control Group at Arcule Inc. Keely has a Bachelor of Science in Chemistry from Merrimack College in Andover, Massachusetts. Thank you for joining us today, Keely.
Thank you, Trevor. Hi, everyone. Again, my name is Keely Murphy, and I'm part of the Strategic Marketing Group here at Thermo Fisher Scientific. Today, I'll be providing an introduction to quantitative analysis using high-resolution accurate math, or more specifically, using our benchtop Orbitrap mass analyzer called the Q-Exactive. In today's presentation, we will discuss what we mean when we say high-resolution accurate mass. We'll cover some of the major differences between a high-resolution approach and a traditional MSS experiment on a triple quad type instrument. We'll highlight some of the benefits that you can gain by going to a high-resolution approach for your quantitative analysis. Then we'll look at the system hardware and experimental design for the data that we'll look at here today.
Keely, I'm sorry to interrupt you, but we're having some problems with your sound. Could we get you to speak up a little bit more, please?
Sure, I'm actually speaking fairly loudly, but okay. Is that any better?
That sounds good.
Okay. Okay, before I cover the high-resolution part of the presentation, I'd like to briefly go over a typical MSS experiment using a triple quadrupole type instrument. In this experiment, your ions are generated at the source, transferred to your Q1 mass analyzer. Here, ions are filtered out based on the mass to charge of your target analyte. From there, your ions are moved to the Q2 collision cell, where a specific voltage is applied to generate your fragment ions. These fragment ions are then moved to the Q3 analyzer, where they're filtered again based on a specific mass to charge for a fragment related to your target analyte. In this type of experiment, this is where you gain your selectivity and quantitative capabilities, especially at low concentrations, by removing a lot of the background noise. But the caveat here is that you need to know before your analytical run the best fragment to use for your target analyte, as well as the optimal collision energy to provide maximum signal intensity. So there is a tuning step generally that's involved before your analytical analysis. From your Q3, your ions are directed to your detector, where they strike a plate or an electron multiplier, generate a current, which is then converted to your mass spectrum. So what do we mean when we say resolution and mass accuracy? Resolution can simply be defined as your target mass divided by the width of your spectral peak at half of its maximum. In short, the narrower the spectral peak, the higher the resolution. The higher the resolution, the more able you are to differentiate between ions with very small differences in their exact mass. For our Q-exactive series, we generally run our quantitative experiments at a resolution setting of 70,000 or higher. It's important to note because this resolution setting is typically higher than what you're capable of on other high-resolution instrumentations, such as a TOF. When we talk about mass accuracy, we're defining the difference between the theoretical exact mass of your target analyte and what you observe on your instrument. This is typically defined in ppm or parts per million and can be calculated by dividing the difference between your experimental and your observed by your target mass and multiplying by 1 million. On the Q-exactive series, we specify a mass accuracy of 3 ppm or less for every scan. This high degree of mass accuracy adds an extra degree of confidence to the data that you collect in high-resolution quantitation. Now let's talk a little bit how we detect ions using an Orbitrap mass analyzer. Unlike a beam instrument like a triple quadrupole, where ions directly strike a plate or electron multiplier to generate a current, with our Orbitrap mass analyzer, ions are collected and introduced into the Orbitrap cell. Once they are introduced, they begin to oscillate back and forth across the inner electrode. Over time, the ions break up into specific bands based on their mass to charge, with the heavier ions oscillating at a lower frequency and the lighter ions oscillating at a higher frequency. As these ions oscillate back and forth, they generate a current, which is measured and recorded, then converted using Fourier transform to generate our mass spectra. There are 3 important things to remember when detecting ions using an Orbitrap mass analyzer. The first is that the Orbitrap can detect and measure multiple ions simultaneously. Second is that it is a very sensitive detector. We are able to measure and record ions as they oscillate back and forth, even at very low population levels. And third is that we're able to measure low population levels of these ions even in the presence of much more intense ion populations. So the dynamic range of the detector is very high. Therefore, you do not need to worry about excluding or filtering out all of your interference ions because the Orbitrap is capable of analyzing them all together at the same time. Here we see a schematic of our Q-exactive focus system. And while the system is capable of many different scan types, today we will focus on our targeted SIM experiment, or selected ion monitoring. In this experiment, ions are generated at the source and transferred to your quadrupole. It's important to note that the system is designed for maximum transmission from your source to your quadrupole, so there's no need to tune any of the voltages for any of the lenses up front, so there's no pre-run tuning required. Once at the quadrupole, your ions are filtered by your isolation window. This is a user-definable setting where the operator can select the mass range that is allowed to pass through the quadrupole. The setting can be as high as 10 AMU in our SIM experiment, all the way down to 0.4 AMU. The ions that are selected to pass through the quadrupole are then collected in the C-trap and then injected into the Orbitrap mass analyzer for analysis. It's important to note here that there is no fragmentation involved. Simultaneous detection of the Orbitrap mass analyzer, along with the high resolution, allows us to remove background and interference ions for our target analyte without the need for MS-MS fragmentation. Therefore, there's no optimization step or tuning step needed before your analytical run. So what are some of the benefits of using a high-resolution, accurate mass approach for your quantitation? Well, the first and most obvious is that there's minimal method development. Because we're not running any fragmentation for this type of experiment, we don't need to prepare a neat solution and infuse prior to our experiment in order to determine the optimal fragmentation and collision energy needed for the analysis. Also, there are no additional lenses to tune. So really, to set up an experiment, all you need is the mass of your target analyte. Additionally, because the Orbitrap mass analyzer is capable of detecting multiple ions simultaneously, you have built-in troubleshooting. Couple that with a 10 AMU isolation window, you're able to identify and characterize nearby interferences to your analyte. From that point, determine whether you need to modify your sample preparation or change your chromatographic conditions. And finally, it's important to note that this is a complementary technique. This adds information to your existing workflow through orthogonal analysis. What we're finding in a lot of labs that have already adopted this technology into their quantitation workflows is that they're moving assays from triple quad systems that they're having trouble with, running some method development on their Q-Exactive system, figuring out the problem, and then deciding whether or not they can address the issue and transfer them back to their triple quad, or if a high-resolution approach is the best way to solve the problem. If it is, then the assays generally stay on the Q-Exactive system for all quantitation going forward. So it really does add to the capabilities that you already have in your laboratory. Here we have the system setup that we use to generate the data that we'll see here today. We have our Q-Exactive Focus and our Vanquish UHPLC system. Our Vanquish system is our next generation UHPLC. It has an upper operating pressure limit of 1500 bar, which allows you to really push your chromatographic resolution, sample separation, faster and more efficiently. It's a very robust, flexible system that's been fully integrated to operate seamlessly with all thermoscientific mass spectrometers. Our mass spec system, the Q-Exactive Focus, is the latest addition to the Q-Exactive portfolio. This system was designed specifically for routine analysis. It has an upper resolving power of 70,000, capable of positive-negative switching, and has a simplified user interface. When setting up methods, the experiments that are available to the user have been greatly simplified, so almost no user input is necessary except for the mass of the target analyte. This makes setup much more efficient and easier, especially from a training perspective. For the data that we're going to examine today, we had a very straightforward experimental setup. We took a group of 7 compounds, created calibration curves using crashed rat plasma to dilute our samples. We analyzed our compounds using a 100 millimeter Hypersil Gold AQ column with 5 microliter injections. Sample concentrations were run in replicates of n equals 6. And for our mass spec method, we used a timed sim approach. So we were able to designate the isolation window specific to our target analytes as they eluted off the column. And this allowed us to maximize the scanning efficiency of the instrument. Now, from the LCMS method perspective, while we did optimize the LC method to provide us with separation for all of our components in our mixture, on the mass spec side, for our source settings, we simply applied default values based on our LC flow rate. So to set this method up, all we needed to enter were our default values for the source settings, are the mass of our target analytes, and their respective retention times. So setup for this method was very generic, very straightforward, very easy to do. Here we see the analytes that we analyzed for this experiment. 6 out of the 7 have deuterated internal standards, and we tried to select a range of compounds that had varying chemical structures, different retention times, and different molecular weights. Again, our samples were analyzed in replicates of n equals 6. Here we see a representative chromatogram containing all 7 compounds. See that we have a nice spread across our retention time, ranging from 1.2 minutes all the way to about 5.5. See that all of our compounds are chromatographically resolved, and we have excellent peak shape for all of our components. So let's take a look at our quantitative scorecard. The goal of this experiment was to determine how well we could quantitate this mix of components in a biological matrices. And if we look at our LOQs, we can see that we're between 5 and 10 picograms per mil for all of the compounds. This means that we were injecting, with our 5 microliter injection, between 25 and 50 femtograms on the column. We also were able to achieve 4 orders of magnitude or greater for all of the compounds we analyzed, meaning we had a very wide linear dynamic range for the working concentration of this assay. If you look at this data, you'll find that it's very comparable to what you would get on a high-performance triple quad mass spectrometer. So by taking a high-resolution approach, you do not have to sacrifice anything from a pure quantitative perspective. So let's look at some specific examples. Here we see oxycodone at our LOQ of 5 picograms per mil. See that our replicates are less than 10%, right? The RSD all the way across the range, and for most they're less than 5%. If we look at our peak shape, we see we have a very nice peak shape at the LOQ, but what stands out to me is how quiet the baseline is here. There's almost no noise down at the LOQ, so we don't need to dig our signal out of a very noisy baseline. This simplifies peak integration and adds confidence to our data analysis. When we dig down into the chromatogram, we can see that even at this very narrow peak width of about 3 seconds at the base, we're still able to get about 10 plus scans across our peak, which is more than enough to provide good reproducibility, especially at our LOQ. See that we're linear across the working range of the assay. We look at the spectra, we see here that although our target analyte is not the most intense ion in the scan, we still have excellent mass accuracy, and at our resolution setting of 70,000, we're fully resolved from all nearby interferences. Here we see buprenorphine with our LOQ of 10 picograms per mil. We can see again that our replicates are all very tight across each calibration point, 5% or less. And again, we have a very quiet baseline, almost no noise for this particular compound. Again, if we look under the chromatographic peak, see that we have plenty of scans and good reproducibility. When we look at the spectra for this compound, we see that in this case, this is the most intense ion in the scan. Now, as I mentioned, we set this up using generic mass spec conditions. We did not optimize the source, gas, heat settings for the mass spec. But I believe that if we did, we could actually get better than the results we're displaying here. But for this particular experiment, we wanted a generic approach just to see how well we could do for this particular mix of compounds. Here we see our last example, clonazepam. Again, we're at 5 picograms per mil. We have excellent reproducibility across our replicates. And we also have a very quiet baseline in the chromatogram. Again, at this very narrow peak width of about 3 seconds, we have plenty of scans across our peak, which is indicated again by our low RSDs across our replicates. For this particular spectrum, you'll see that there are a lot more interferences here. And in fact, our target analyte is very low compared to some of the other ions present. But still, we're able to obtain very good mass accuracy, right, and reproducibility at the low level. So this really does indicate that the Orbitrap mass analyzer is very capable as a sensitive detector with a very high dynamic range. We wanted to address injection-to-injection reproducibility. So we looked at the raw area of some of our internal standards. Here we see buprenorphine, very low variability across the injection series. These are all the injections for our experiment, CV of less than 4%. We look at clonazepam, we see something very similar. We see a CV of less than 3%, very reproducible all across our injections. This adds another degree of confidence to the data that we've collected. We also wanted to look at chromatographic reproducibility across our replicates. Here we can see buprenorphine at our LOQ of 10 picograms per mil. See that all of the peaks are very smooth and reproducible. Important to note that no smoothing was applied here. Okay, we also mentioned mass accuracy. If we look here at buprenorphine at our 10 picogram per mil LOQ, we can see we've labeled every scan with its delta ppm. See that even at the shoulders, we're below the 3 ppm spec. This adds another degree of confidence to our data and also allows us to apply very tight mass extraction windows. This helps us eliminate noise while adding confidence to our data set. Now, before I sum up, I'd like to quickly highlight the performance of our Vanquish UHPLC system. We can see that all of our compounds are chromatographically resolved, but these 5 components come out in just about 1 minute. We actually ran this experiment on our previous generation LC systems, and we weren't able to separate them. So with the Vanquish system, we wanted to try to improve the cycle time efficiency of the Q-Exactive by timing some of our transitions. But in order to do this, we needed to make sure that we had reproducible retention times to avoid having our target peak shift out of its acquisition window. So here we've plotted the retention time over the course of the injections. And what we see is that we have very tight CVs with a standard deviation of about a half a second all across this assay. So the Vanquish is able to deliver very reproducible retention times, which actually enhances our ability to analyze our compounds on the mass spec. In summary, the Q-Exactive, more specifically the Q-Exactive Focus, is being used in many different application areas and many different labs. Today, we've highlighted the capabilities for quantitation for the Q-Exactive Focus using SIM, highlighted some of the reductions that are possible using the SIM mode, right? There's no tuning involved. We also have built-in troubleshooting, and I think that we've demonstrated that this is a highly sensitive system that is very capable of high-performance quantitation. Really, this is a tool that will add value to an existing quantitation workflow, and it actually adds. And instead of just putting in another box that does the same thing, this will add a complementary technique to what you already have in place. If you have any questions about the data that I presented here or any of the applications, please feel free to contact me at the email below. Thank you for your time.
Well, thank you very much. And thank you all for sending in your questions. I encourage you to continue to do this throughout the presentations. If you're joining us late, you may ask questions simply by typing your query into the question box, which is located at the bottom of your screen. Our next speaker today joins us from Neoterix. Dr. Stuart Cushing currently holds the position of Senior Research Scientist at Neoterix, a company dedicated to developing novel microsampling solutions for the pharmaceutical and clinical markets. Dr. Cushing is a physical organic chemist with over 10 years of experience developing products for the direct detection and analysis of targets, including viral, bacterial, and protein pathogens, as well as small molecules that serve as diagnostic markers for disease states. His publication set has increased the understanding of the thermodynamics and kinetics of the binding of small molecules and biological molecules, and illustrates the role of those properties in the development of detection systems. Previously, Dr. Cushion served as Senior Research Scientist at Phenomenex, where he played a key role in the development of novel sorbents for chromatography and a line of phospholipid removal and protein precipitation products. Thank you for joining us today, Stuart.
Good morning. So today I'm going to talk a little bit about some advances in microsampling for in vivo and DMPK studies. So our agenda for today, first I'm going to speak briefly about blood microsampling. Then I'm going to move on to some details about dry blood spotting cards, which is a technique from the 1960s that's widely used for taking microsamples for neonatal screening. Then I'm going to speak a little bit about volumetric absorptive microsampling, which is a novel approach to microsampling. Then finally I'm going to close with a mouse PK study using volumetric absorptive microsampling. So what is blood microsampling? Well, it's not easy to find a definition for this if you go out and look into the journals. But for our purposes here, we're going to describe it as collecting reduced volumes of blood from animals or humans with the purpose of providing a result. Now, in some cases, the result will be a qualitative result, and in others, it'll be a quantitative result. So in other words, in traditional workflows, you're going to be collecting much greater than 200 microliters of blood. But in a microsampling workflow, you're going to be collecting much less than 100 microliters of blood. Now, one of the main advantages of microsampling is animal welfare. This is important, especially in the context of mice, where blood volumes are quite limited. It allows you to sample multiple time points from the same animal and significantly reduce the number of animals that are needed. This serial sampling allows you to combine PK/PD and efficacy assessments in the same animal, and then depending on the workflow, you can reduce or eliminate inter-animal variability, which will improve data quality. And then the bottom line is that it leads to cost savings and time savings. There's going to be less spent on animal models and the disposable costs for them, and then there's going to be reduced labor and animal handling requirements. Now I'm going to focus a little bit about dry blood spotting. I'm going to talk a little bit about dry blood spots. The benefits of dry blood spots are that they are an easy sample collection and storage tool. It allows for simple shipping as well because it is a dried matrix approach. There are no plasma processing steps, and what that means is there's no centrifugation that's required or aliquot generation and all the relabeling that comes with that that is found in a traditional whole blood plasma workflow. There's no dry ice or couriers or freezers. Those things just simply aren't required because it is a dried matrix approach. Blood is spotted onto the card. It is then dried, shipped to a central lab for testing where the card is then punched with a hole puncher, and then that punch is analyzed. But this leads into one of the primary limitations of DBS cards, and that is that the sub-punch workflow is very laborious. It's manual. In most labs that we go to, we see people actually working with hole punch devices. Some people have set up some complicated apparatuses to make it easier, but overall it's quite difficult. Additionally, there's a substantial amount of blood dust that is created whenever you're generating a sub-punch. And then the generation of the sub-punch itself leads to something known as the volumetric hematocrit bias or the volumetric hematocrit effect. Another limitation that's not shown here is the chromatographic nature of the paper itself. So the blood spot that is collected is not actually uniform. So when you take a sub-punch, you may be getting various parts of the blood. So next I'm going to explain the volumetric hematocrit effect that is found in DBS cards. So the viscosity of blood has a positive correlation with the hematocrit or the packed cell volume of the blood. What this means is that the lower the hematocrit of the blood, the more that a blood sample will spread out on a DBS card. So then when you take a uniform sub-punch from the card, the volume that's contained in that sub-punch is going to be a function of the hematocrit of the blood. And this leads to what we see below here. Now this is acetaminophen analyzed using an FTA elute card. And what you can see is that there is a positive bias at high hematocrit and a negative bias at low hematocrit. And this is in part due to the volumetric hematocrit effect. So now I'm going to switch gears and I'm going to talk a little bit about a novel device that overcomes the deficiency of DBS approaches, and that is volumetric absorptive microsampling. It uses capillary action and a highly controlled internal porous volume to collect an accurate and precise volume of blood. In this case, it's around 10 microliters. It's available in a high throughput and automatable format. So what's shown below here is a graphic of the product that is branded, the Mitra microsampler, that uses the volumetric absorptive microsampling approach. The first feature of it that I would like to talk about is that the distal end fits most standard 20 to 200 microliter pipetters. So there's a hole that is present there, and that hole can be adapted to liquid handlers like a T-can or a Hamilton. Then there is a barrel that is easily written on or labeled with a wrapped label. There are some ribbed features that allow you to simply drop this entire apparatus into a collection plate for analysis. And then finally, what's shown in red at the bottom of the device is the collection end of the device. So now I'm going to show a brief video of the device in action. What you're going to see is a plastic stick with a white tip on the end of it. And when it contacts the blood, you can see it turn red in just about 2 seconds. So that is the collection event. You simply take the white tip and touch it to the blood source, and you get an accurate 10 microliters of volume. So now I'm going to move on. So the data shown here shows that the Mitra microsampler has no volumetric hematocrit bias. This was collected courtesy of Neil Spooner and Phil Denniff at GSK in the UK. And what you can see from this data is that the maximum volume bias from 45% hematocrit is about 3%. In fact, over the entire set of data, there's only a 5% volume deviation across a hematocrit range of 20% to 70%. And that actually is approximately the stated precision of the Mitra microsampler, which is around 4%. That's the type of accuracy, sorry, that's the type of precision, the RSD, that you will generate using this device. The workflow for volumetric absorptive microsampling is actually quite similar to DBS workflows. You collect a sample, then you will dry it for more than 2 hours. You'll then take the device and you can then store it or transport it at your leisure. Then, when you're ready for extraction, you can just extract it with common solvents. What's shown here is a 100% methanol. You would take that methanol, you would just add it to the collection plate, drop the microsampler into the collection plate, sonicate it for 15 minutes, and then vortex it for 60 minutes. You then would remove the sampler. You can then transfer the extract, dry it, and reconstitute it in initial mobile phase for analysis. Now, prior to the launch of the Mitra microsampler, we performed a pilot study with 8 different pharmaceutical sites. And I'm going to present an assay performance evaluation using volumetric absorptive microsampling that was performed by Bristol Myers Squibb. This is courtesy of the lab of Mark Arnold and one of his senior scientists, Chin Ji. So what's shown here is an extraction methodology. What they did was they simply shucked the tip, they removed the tip into a 96 well plate. They then added buffer, so 75 microliters of water with ammonium acetate and acetic acid. The tip was then sonicated for 10 minutes. They then added 25 microliters of their internal standard and vortexed the plate for 10 minutes and then centrifuged it briefly. To that, they then added ethyl acetate and hexane, 70-30 to all the samples. It was shaken on a linear shaker, centrifuged, and then the supernatant was drawn off, dried down, and then reconstituted for analysis. This approach essentially boils down to a reconstitution of the whole blood off of the tip with water, followed by a traditional liquid-liquid extraction approach. And the primary reason why this site used this approach is because liquid-liquid extraction is not a particularly selective sample preparation approach, and they were looking to analyze four different analytes using the exact same extraction methodology. This approach, this extraction approach led to an absolute recovery of greater than 85% for all 4 of the analytes. And what's shown here is the assay bias evaluation for all 4 of the analytes as a function of hematocrit. And what you can see is that their optimized extraction completely eliminates the hematocrit bias for all 4 of their analytes. Now, I can't share with you what the analytes are here because they were proprietary. They continued with this study, and they showed that the analytes were stable. The test, the entire workflow, and the signals that were generated by the analytes were stable for 30 days. So these are the benefits of using an optimized extraction methodology. You can eliminate hematocrit bias, and you can also ensure that your assay will be stable. Now I'm going to move into a mouse PK study. This study was performed by Sci Analytical Strategies in San Diego, and it's courtesy of Dan Castle there. This is the study design for the mouse PK study. An IV dosing route was used. The test article was acetaminophen at 2 milligrams per kilogram into the tail vein. Now there were 6 different time points that were used. There was a pre-dose, so there were a total of 7 time points that were used for the study. The study design used cardiac puncture of 3 different mice at each time point. So there were a total of 21 mice that were sacrificed over the course of this study. And then the comparison that was being done was a comparison of a traditional whole blood plasma workflow to a meter microsampling dry whole blood approach. What's shown here are the 2 different extraction approaches. They are essentially both protein precipitation methodologies. So with the whole blood plasma approach, there was a 9-to-1 protein precipitation that was used with methanol. So the approach here was to just simply spike in 9 parts methanol for each 1 part of plasma, and then to vortex for 30 minutes. The meter microsampler also used a protein precipitation method, although, as you can see, it is a little bit more complicated here. We recommend significant agitation because it is a 3-dimensional object that you are extracting from, so it is a solid liquid extraction that you're performing. So in this case, 100 microliters of water with 1% formic acid was added to the meter microsampler. In this case, they vortexed for 30 minutes, and what this does is it reconstitutes the blood. Very similar approach that was shown earlier with the liquid-liquid extraction. It began with a reconstitution of the whole blood, and then there was a partial precipitation event that was done. So 100 microliters of methanol was added. It was vortexed for 30 minutes, and then finally the tips were removed, and 600 microliters of cold acetonitrile was used to complete the precipitation with a vortexing step of 10 minutes. Now then, with both approaches, the sample was then centrifuged in order to remove the precipitate, and then the supernatant was pulled, transferred, and then evaporated and reconstituted for injection. These are the 2 standard curves for this mouse PK study. On the left side, this is a whole blood plasma approach, so that's the traditional whole blood approach. And then on the right side, you can see the standard curve using the meter microsampler. You can see that the R-squareds in this case for the calibration curves were above 0.99. And then here are the results of the study. So the PK parameters are shown. This is a concentration versus time profile for acetaminophen. And you can see that there's a rapid decline in the concentration over the period of about 1 hour. The PK parameters are very similar. The area under the curve, along with the systemic clearance and the volume of distribution, are all quite similar to each other. The only parameter that's shown to be different here is the terminal elimination half-life, so that's 0.6 for the whole blood plasma study and 0.2 using the meter microsampler. But the overall interpretation of this study would be the same using either workflow, and that is that it has moderate to high clearance and then a relatively short half-life. Now, conveniently, acetaminophen is a drug that has a blood cell to plasma distribution of 1-to-1. So this is the type of the result that you may expect when doing this sort of comparison, because a traditional whole blood plasma workflow actually throws out all of the blood cells. So if this distribution weren't 1-to-1, then you might expect some deviation based on that. And then here are the study conclusions. So the meter microsampler allows for hematocrit-independent collection of a volumetrically accurate 10 microliter blood sample. And what that does is it makes it possible to conduct full PK time course profiles. And it opens the door to combining PK/PD and efficacy assessments in single animals. We've just shown a PK study where the data from a traditional whole blood plasma approach is concordant with a dried matrix approach using the meter microsampler. And then finally, microsampling will allow for significant reduction in animal usage and handling costs. So I would like to thank you for your time. This product was released last June, and there are, as you can imagine, there's quite a bit of interest in the product. And there are already a number of publications that are in print, and there are many more coming. So I'd like to thank you for your time, and I'm happy to field any questions that you may have about volumetric absorptive microsampling.
Great, thank you very much, Stuart. And just a reminder that all of our speakers today have kindly provided additional resources. These can be accessed via the resource widget, which is located at the bottom of your screen. I believe if you're both ready, we'll move directly into the question and answer session. We do have a number of questions that have come in from the audience. And Keely, maybe I can start with you. You mentioned that there was no smoothing applied to your peaks. Can you briefly go over smoothing and its application?
Sure. Smoothing is generally used to account for variability in your sampling across your chromatographic peak. Generally, we do not apply smoothing with our high-resolution accurate mass data because we do not need to. And depending on the lab and their particular SOP, some labs allow for it and some do not. So if we can avoid it, we do.
Great, thank you. Stuart, could you go into a little more detail on how the Mitra microsampling is compatible with liquid handlers?
Well, as you probably well know, liquid handler vendors would all prefer that pipette tips can only be used on their liquid handler. So there are always fitment issues with every liquid handler that exists. So we are currently working in collaboration with a number of liquid handler vendors in order to make the Mitra microsampler compatible with liquid handlers that are on the market. Essentially, if you saw the design, it looks very similar to a pipette tip, and that's intentional so that it can just simply fit onto a liquid handler.
Great, thank you. Keely, why is mass accuracy important, and how often do you need to calibrate this system?
Good question. The mass accuracy is important because it allows you to obtain a very high degree of confidence for the data that you're collecting. By using or by having a good mass accuracy, you're able to really narrow the mass range that you use to generate your chromatograms. This will improve data confidence, it will eliminate noise, and it really helps you to be sure that the signal that you're integrating as part of your chromatogram is due to your analyte of interest and not due to some background noise or other interference. Mass accuracy can also be used as a tool to qualify your data. If you see scans falling out of the specified mass accuracy range greater than 3 ppm, then it can be an indicator that you have an interference that needs to be addressed in your assay. So you can use it as a validation tool. As far as instrument calibration, we recommend that the system be calibrated at least every 48 hours. But what we find in practice is that the system calibration will last for up to 5 full days and will maintain that 3 ppm mass accuracy. And this is a lot different from other high-resolution instruments where you have to recalibrate the system very frequently, and for some systems during your actual analytical run. So you're able to calibrate once with an external calibration and maintain your level of mass accuracy throughout the course of your entire analytical run, even if it's for an extended period of time for 1 or 2 days.
Great, thank you. Stuart, acetaminophen has a very low log P and is very soluble in water and methanol. How do you handle compounds with a higher log P?
So what we're talking about here are analytes like vitamin D or THC. So in that context, you can imagine that what you would be doing instead of doing an extraction with just pure water would be either doing a solid liquid extraction using a more nonpolar solvent. An example there would be something like 50-50 ethyl acetate or methanol. Alternatively, if you saw the liquid-liquid extraction approach that was used by Bristol-Myers Squibb, you can see that what they're using there is a mixture of ethyl acetate and hexane for their liquid-liquid extraction. That's also a fairly nonpolar solvent mix. So, from general chemistry, like dissolves like, and so the approach to extraction of hydrophobic analytes is generally the use of a more hydrophobic solvent, either through a solid liquid extraction approach or a liquid-liquid extraction approach. We've actually seen quite a bit of success using protein precipitation strategies as well, assuming the appropriate modifiers are used. So in that case, you often need to use a formic acid modifier. But all of those approaches are fairly effective.
Okay, thank you. Keely, can the Q-Exactive Focus run MSS experiments as well?
Yes. There is a collision cell, and the ions are transferred to the collision cell before they're analyzed in the Orbitrap mass analyzer. Typically, for our quantitation experiments that we run, we do not need MSS unless we have a very complex sample matrix. So for the most part, we achieve great quantitative results with just our sim experiments. But when you do have more complicated samples, the option is there, and you can run a typical MSS experiment. The main difference between an MSS experiment on the Q-Exactive and a traditional triple quad instrument is that on the triple quad instrument, you need to know beforehand the mass of your fragment. Where on the Q-Exactive, after your fragmentation, the Orbitrap will be analyzing all your ions simultaneously because there's no additional filtering step. So you don't need to know what your fragment is going to be beforehand because the Orbitrap will detect it regardless. But yes, it is capable of MSS experiments, and it's there if you need it.
Great, thank you. Stuart, what is the typical reduction in the number of animals required for a study when using a microsampling workflow versus a traditional plasma workflow?
Well, that really depends on what the initial workflow was. In a lot of cases, especially if the animal model that is being used is an expensive one, the workflow that is currently being used will be using a small number of animals to begin with, maybe only 3 per time point. 3 is a number that we see quite a bit, but sometimes you will see 6 animals that are sacrificed at each time point. So the reduction is normally either a third or a sixth. Those are the most common that we've seen.
Great, thank you. Well, gentlemen, we've come to the end of our questions, so I'd like to thank you for your discussion. And this brings us to the end of today's webinar. If you have any further questions, please consider reaching out to our panelists directly, as they are the true experts in the field. Their contact information, as you can see, is available on the screen now. And just a reminder that today's webinar video will be available at the link you see on the screen. On behalf of Lab Manager, I'd like to thank our panelists today for all their hard work that they put into their presentations. And I would like to thank our sponsors, Thermo Fisher Scientific and Neoteryx, for supporting our Tech Trends webinar series. As well, I would like to thank all of you for taking time out of your busy schedules to join us today. Please mark your calendar for our next Tech Trends webinar, which will focus on innovations in microplate technology. This webinar will be taking place Thursday, May 21st, from noon until 1:30 p.m. Eastern Time. And for more information, please visit our website at www.labmanager.com. We hope you can join us again next time. Thank you, and enjoy the remainder of your day.